We've already paid a lot of attention to the molecular structure of DNA. In fact, right depicted in front of us, we have two strands of DNA forming a double helix. And we can look at the telltale signs that this is DNA. And in particular, we can look at the 5-carbon sugar on its backbone. We see, and let's actually number the carbons. This is 1', 2', 3', 4', 5'. We can see on the 2' carbon we don't have an oxygen attached to it. We don't have a hydroxyl group attached to it. And because of that, we know that this is not ribose, this is deoxyribose. This right over here is deoxyribose, and these two are also deoxyribose. So that tells us that we have two strands of DNA, deoxyribonucleic acid. So let me write this down. This part of the chain, this is derived from a deoxyribose being attached to phosphate groups and a nitrogenous base. So deoxyribose. So what would we have to do if we wanted, instead of viewing this as a as two strands of DNA in a double helix formation, well, how would we have to rearrange, how would we have to edit the left-hand strand if instead we wanted to imagine that the left-hand strand is, say, messenger RNA being generated during transcription with a single strand of DNA here on the right? Well, to turn this into RNA, or to make it look like RNA, on the 2' carbon, well, we want to turn the deoxyribose into just ribose, so we would want to add a hydroxyl group right over here. So I add a hydroxyl group over there. Actually, let me do that. Do the hydrogens in white. So add one hydroxyl group there, and I want to do it on all the sugars on the left strand's backbone. If I want this to be a single strand of RNA, and RNA tends to be single-stranded. So oxygen, and then a hydrogen. And so this hydroxyl, adding this hydroxyl group, instead of just having another hydrogen, just a hydrogen by itself over there, this tells us that this sugar is no longer deoxyribose, this is ribose. So now we have ribose. We now have ribose in our backbone, which is a telltale sign that, well, at least now we have the backbone of RNA, ribonucleic acid, versus DNA, deoxyribonucleic acid. Now, you might think we're done, but we're not quite done, because the nitrogenous bases on RNA are slightly different than the nitrogenous bases on DNA. On DNA, your nitrogenous bases are adenine, guanine, and adenine and guanine are the 2-ringed nitrogenous bases right over here. This is adenine, this is guanine. And you also have cytosine. Cytosine, I'm going to do these all in different colors. Cytosine and cytosine and thymine. I'm getting to the punchline too fast. And this right over here is cytosine, and this is thymine. And cytosine and thymine are single-ringed nitrogenous bases. We call them pyrimidines. Adenine and guanine, we call them purines. This is a little bit of review. In RNA, you still have adenine, you still have guanine, you still have cytosine, but instead of thymine, you have a very close relative of thymine, and that is uracil. So the way that this is drawn right now, this nitrogenous base, remember when we started this video, it was double-stranded DNA. This nitrogenous base right over here is thymine, and it forms hydrogen bonds with adenine right over here. If I want to turn into uracil, I just have to get rid of this methyl group right over here. So if I just do this, and if I were to replace it with a hydrogen that is just implicitly bonded there, well, now I'm dealing with uracil. So you see that uracil and thymine are very close molecules, or they're very similar nitrogenous bases, and that's why they can play a very similar role. And it's still the case. And so what uracil pairs with, it pairs still with adenine, the same thing that thymine pairs with, and everything else is, of course, still the same. Now, an interesting question is, why uracil? Why not thymine? Or you could say, why thymine? Why not uracil? And based on what I've read, it actually turns out that uracil is a little bit more error prone. It might be able to bond with other things when you're coding. It's a little bit less stable than thymine. And so uracil makes the RNA molecule, or actually makes the machinery of information transfer, it makes it less stable. It's a less stable, I guess, way to transfer information. And based on what I've read, in evolutionary history, RNA molecules, most people believe, predate DNA molecules. And then so in the early stages, you had a lot of change, and so uracil molecules were just fine, and there was a lot of errors and whatever else. But then once you had, I guess, information needed to be a little bit more persistent and a little less error prone, well, then thymine helped stabilize. Thymine helped stabilize things. There's also the view of, well, why has uracil stuck around? Well, RNA molecules, they have all of these roles in cells. Messenger RNA molecules are taking information from the DNA and getting it transcribed or getting it translated at the ribosome. But they shouldn't hang out forever. That you actually want them to be somewhat unstable. So it's an interesting question to think about. Why do we have uracil instead of thymine? Or why do we have thymine instead of uracil? But this is one of the telltale signs of that we are now dealing with an RNA molecule. So now what we have on the left-hand side, now all of this business, actually let me do this in a different color. All of this business, this strand right over here, we can now, the way it's drawn, we can now consider this an RNA molecule. And if we assume that this is happening during transcription, when a DNA molecule, where a single strand of DNA would want to replicate its information, then this over here would be mRNA, messenger RNA. And so what's going on here? Well, let's think about it. This one, the way it's the RNA, the messenger RNA, the way it's oriented, we have phosphate group, then we go to 5' carbon, 4', 3', then phosphate group, then 5', 4', 3', then phosphate group. So this is oriented 5' on top, 3' on the bottom. While this DNA molecule is oriented the other way. This is a 5' carbon, this is a 3' carbon. So we have phosphate, 3', 5' phosphate. So we have 3' is on top and 5' is on the bottom. So if we wanted to think about what's happening, maybe using the symbols for the nitrogenous bases, we could say, all right, we have our mRNA molecule here, and this is its 5' end, and this is its 3' end. And then the top nitrogenous base right over here, this is uracil. This is uracil. And then the second one over here, this is cytosine. So this is cytosine. This is cytosine right over here. And this is being transcribed from a DNA molecule, from this DNA molecule on the right-hand side. So this is DNA. And this DNA has an anti-parallel orientation. It's parallel, but it's kind of flipped over. The sugars are pointed in a different direction. So this is going from, this is the 3' end, this is the 5' end. And we see that the uracil is hydrogen bonded to adenine right over here. So adenine, and I'll draw dotted lines to show the hydrogen bonds, and that the cytosine is hydrogen bonded to guanine. So this right over here, that is guanine. And actually, I'll do the hydrogen bonds in white. So, they are, actually there's multiple hydrogen bonds going on here. But just to be clear, this is mRNA, and on the right we have DNA, and this could be happening during transcription. This could be happening during I'm having trouble changing colors. This could be happening during transcription. Now, what are the types of RNAs out there? And we've talked about this in other videos. Well, you have messenger RNA, which is an important role in taking information from DNA and getting it eventually translated with the help of tRNAs and ribosomes. And though I've just mentioned another type of RNA, and that's transfer RNA. So transfer RNA, tRNA. And in the video, the overview video on transcription and translation, we talk about how tRNA does this, but it brings amino acids it has amino acids attached at one end, and then it has anticodons on the other end that essentially pair with codon fragment or codons on the mRNA, and then that allows it to construct proteins. And this actually is this right over here is a visualization of a tRNA molecule. So a lot of times when we think about DNA, we think about, okay, mRNA or RNA is an intermediary to be able to eventually translate it into proteins, and that is often the case. But sometimes you also just want the RNA itself. The RNA itself plays a role in the cell beyond just transmitting information, and that's an example here with tRNA. And you can see its interesting configuration, where the amino acid will attach roughly in that area up there, and then you see the anticodon right down here in the bottom right. And different tRNA molecules will attach to different amino acids, and they'll have different anticodons here. So this is another use for RNA. And then others include ribosomal RNA. And they actually play a structural role in ribosomes, which is where translation occurs. And you also have things called microRNA, which are short chains of RNA, which could be used to regulate the translation of other RNA molecules. So RNA, DNA gets a lot of the attention, but RNA is really, really, really important. And a lot of people believe that RNA came first, and it's potential that the first life or pseudo-life ever was just self-replicating RNA molecules, and that DNA eventually evolved from RNA, but RNA stuck around because it's still very useful.